Good afternoon, everyone, and welcome to the last liver seminar of this season before the holiday break. Today, we have the pleasure to have with us Professor Thomas Baumert. He's right now the director at the Institute of Translational Medicine and Liver Disease at the ANSER in Strasbourg, in the University of Strasbourg. He leads the Laboratory of Excellence, HEPSIS, and he's the chief also of the gastroenterology service at the Strasbourg University Hospital in France, as well as the founder and advisor for Alentis Therapeutics in Switzerland. So Thomas received his medical doctor degree at the University of Heidelberg in Germany, and then he moved to Heidelberg to do this his doctoral thesis at the German Cancer Research Center, the Deutsche Krebs Forschungszentrum in Heidelberg, and his internship in internal medicine at the Ludwig Maximilians University in Munich. And then he moved as a fellow to Harvard Medical School at the Massachusetts General Hospital and the liver disease branch at the NIH in Bethesda. So he came back to Germany and joined the Department of Medicine at the University Hospital in Freiburg as an internist and gastroenterologist, also vet.rai, so associate professor, and established his laboratory focusing on the molecular pathogenesis of viral hepatitis and liver disease. Finally, he relocated to the University of Strasbourg as a full professor of medicine to lead the new research unit created by ANSER and establish a very high recognized program in translational hepatology that he's now leading. He also took an academic year as a research scholar in the Broad Institute of MIT and Harvard in Boston. His research focuses on the modeling and discovery of the cell circuits underlying fibrosis progression to cancer using liver disease as a model. Professor Baumert has received many awards, including the Hans Popper Young Investigator Award, which is very prestigious. He has published over 400 scientific articles, and he has raised a lot of funding, not only from European funds, but also from funds from the United States. So he's a very well-recognized scientist and clinician, and he's an associate editor of the Journal of Hepatology, and belongs to the editorial board of many journals, including gastroenterology and hepatology. So today's talk, it will be about the discovery of novel circuits, and I give the word to Professor Baumert, so you can now unmute and start, Thomas.
Yeah, thank you very much for the kind invitation, Javi. And I'm really happy to give this presentation, although I cannot be in person. But the two of us met at EASL, had a 1-hour conversation, and I thank you again, basically then to come next year also in person to basically, I spared really a lot of other research to show. And I think it's really, a good time to talk about liver disease drug discovery. We have seen a lot of progress. As you know, we all witnessed or participated in the revolution of curing hepatitis C, controlling hepatitis B, and then also advancing in HCC and also MASH fibrosis now moving forward. So today I would like to talk about how we can harness and use prognostic gene signatures from patients to discover liver disease therapeutic and targets. So in my talk today, will talk about the unmet medical need, treatment of liver fibrosis and cancer, then show how we can model liver cell circuits of liver fibrosis progressing to HCC using the CPLS models, which we have been working for the last 10 years. And how can we discover novel liver disease therapeutics and targets of CPLS in patient models? Will you show one example? Here we have many more, and then conclude and perspectives. So I think we all know here, particularly in the audience here, liver fibrosis progressing to cancer, it's a major health burden with absent or unsatisfactory therapeutic options. We have now resmetirom entering the clinic. I will talk about this in a minute, but I think there's really, really much more room to improve. We have a high global prevalence expected to grow. We estimate that about 1 billion of patients have liver disease. Out of the total, we have a prevalence of advanced liver disease fibrosis 1.5 to 6.5 expected to grow. around 3 million patients with advanced fibrosis in the United States. And we all realize that this is the biggest challenge because it's the major cause of HCC. We have now no approved antifibrotic therapy. We have now an approved therapy for MASH, which some efficacy on fibrosis. We have still very limited efficacy, if we are honest, of the compounds, especially on fibrosis. We have been making nice progress on metabolism. And basically what I think we believe is really giving the growing numbers of patients, in particular with MASH and basically still hepatitis C and hepatitis B and also alcoholic liver disease, we have really a very urgent need to prevent also liver cancer. So on this slide, you see, I think this is a slide I like very much, is basically how I see basically the challenge, and I call it the fibrotic cliff. So we have basically here early stage fibrosis, F1 and F2 patients are asymptomatic. They don't have any really clinical problems. They move to F3 and F4, then they move to cirrhosis. We have been learning that basically the pathogen at the end, the clinical features and the clinical manifestations, no matter what it is, alcohol, MASH, HCV or HBV, is very, very similar. We have then liver cirrhosis with decompensation, HCC, liver failure, and death. And the point where we need to intervene is really at this stage here, F3 and F4, to prevent patients falling over the cliff, to prevent them from all the complications, and also at the same time to basically save in the healthcare system, because the treatments of these complications is very, very costly. So this is, again, I just want to highlight, I'm sure all of you know this, but I think it's really, I think liver cancer is one of the, I think, best examples. There are also other cancers, but here we have really the fibrosis is the key risk factor for HCC. Here you see that this very nice study from Sanyal et al. New England Journal, where this really was shown in a large cohort. We have around more than 800 tests annual in rising. And the biggest challenge is that we can early stage, surgeries is able to resect the tumors, but we have unfortunately recurrence in up to 70% in 5 years. And so far, we simply basically are watching. We are now trying with chemotherapeutic approach to prevent the recurrence. There's some good data now also, but still this is a major challenge. And we all know also we have seen tremendous progress with the clinical approval of several new compounds in the last decade. Still, the treatment of advanced HCC remains unsatisfactory with an overall response rate at this point with the standard of care to less than 30%. So there's really much room to improvement. So one question which I think is something which always comes up, and that's also particularly important, I think, for the young medical doctors and also the students. When I basically myself was a student, we were considering fibrosis and advanced fibrosis as something irreversible, and the damage was done, and there's basically nothing you can do about it. You can try to take care of the patients, but there's nothing to do. And that's something I think, especially in the liver, we have learned that we can reverse fibrosis. So in particular in early stages, this has been shown by bariatric surgery, but also very nicely shown by a large series of studies showing that the cure now of hepatitis C virus infection is able really to reverse fibrosis. Same for alcohol withdrawal. We see this in the clinic. But there also seems potentially a point of no return once you have advanced fibrosis, especially if you have cirrhosis. Then even there are also some very nice studies have shown that for hepatitis C, even if you cure the virus, it is too late, and basically the reversal seems not possible anymore. And so this is an example, and I just want to talk about, before I move into the prognostic signature and the drug discovery, I want to share my experience with hepatitis C. As you know, I've been working for more than 25 years on hepatitis C virus. And so I think it's a fascinating virus, which allows us now, after we have discovered and using the cure, still this is a virus where we can learn a lot about liver disease pathogenesis and carcinogenesis. So what we have learned over the last couple of years, and these are the early studies, have been reproduced by several others, that we have still a clinical challenge that the risk of HCC persists following DAA cure in patients with advanced fibrosis, in particular with liver cirrhosis. And this is here this summary of the study from Kanwal, El-Serraj, in this retrospective study of 22,000 patients. They were treated with DAA, where they had 30% of cirrhosis. What we see here, that we see basically an improvement of the risk of HCC with following DAA cure, but we have still a substantial risk of developing cancer even following the cure with direct acting antiviral. So that basically shows there's also a cohort including the HCV treated patients which will need additional treatment to prevent them from liver cancer. And how does that work? And I just want to review some of the studies we have been doing in the last couple of years, and we found first in models, animal models, but also then in different cell-based models, and then also in patients, that the virus leaves genetic footprints in the liver, and these footprints, these changes, they remain following cure. So we have basically we showed that here in 2 publications, and also our colleagues here, group from Israel, showed the similar was reproduced now on several different levels, also with T cells. And here, what we showed that the virus basically results in a substantial modification of the epigenome, which then results in altered gene expression, which then cause liver disease. And what is I think very exciting, but very well, at the same time also very challenging and difficult, is that unfortunately these changes they persist following viral cure, and we believe they continue. These are one of the drivers which drive this dysregulation, which are driving the HCC risk. And we observed that actually these changes are most pronounced in patients with advanced liver disease, and also we see that the reversal only partially occurs in these patients with advanced liver disease. So this is really something related to advanced liver disease, and we believe this will have a causative effect. We showed that also with pharmacological inhibition at that time. And we also found that actually that chronic HCV infection leaves an epigenetic viral footprint in patient livers after DAA cure. So here, this is an example of persistently dysregulated gene expression after viral cure, and this involves several cancer risk genes, and the most prominent dysregulation we found in patients with advanced fibrosis and cirrhosis. And we are currently working now to develop a blood test, which we have a big program with Pierre Naon, and also we're working with Judith Hoshida to develop non-invasive biomarkers to identify patients at risk. And these data here have been very helpful to identify candidate sets of genes and proteins. So I want to now switch gears and basically show you how we believe one aspect, how these epigenetic changes at the end induce the cancer risk. And I think this is a really very fascinating story. We have been working over the last couple of years. And this is basically, we looked at how hepatitis C virus infection changes the liver circadian clock. And so I think, as you all know, the liver circadian clock is a critical regulator of liver metabolism and many cellular processes. You've seen this here in this cartoon, starting from xenobiotic detoxification, regulation of nuclear receptors, cellular processes, glucose, carbohydrate metabolism, amino acid metabolism, lipid and bile acid metabolism. And there have been several publications just in the last couple of years showing nicely that the perturbation of the circadian clock is actually associated with the development of metabolic changes in MASH, and also they're increasing cancer risk. So we wanted to know now, what about the hepatitis C virus? We want to understand the functional role of the circadian clock in HCV infection and understand whether the virus dysregulates the clock and whether this dysregulation can contribute to the risk of patients developing HCC. So just to give you a very brief overview about the circadian clock, this is basically, it is mastered by a series of key regulators shown here by BAML1, clock, RevUp alpha, This consists of a transcriptional translational feedback loop, which is present in every cell, including the hepatocytes. We actually just have a paper in Journal of Hepatology and Revision. We also show this is actually also present in stellate cells and myofibroblasts, where the circadian clock can drive liver fibrosis. These genes, they result in the rhythmic control of gene expression. This is time of day dependent. They also regulate hemostatic functions by transcriptional and post-transcriptional mechanisms. So this has been characterized in detail in the last couple of years. And we are also trying, and others trying, to use the circadian clock as a therapeutic target to treat metabolic disease and also fibrosis. And so here, what we showed here is basically what we did. We used the human liver chimeric mouse model. As you know, this is the only in vivo model infectable with hepatitis C virus aside from the chimpanzee. And these mice are transplanted with human hepatocytes. These are human UPS kid mice, so they have a deficit in liver regeneration, so they are able to, they're permissive to engraftment of human hepatocytes. And then we are able to infect these mice with hepatitis C virus for many, many weeks, many months, actually. And what we did here, we infected them for 8 weeks. And then what we did, we harvested, this is really it's very hard for the people doing this, basically. Then we harvested the livers over a time period of 24 hours at different time points, time point 0, 4, 8, 12, 16, 20. And so what you see here is basically as an example of here the clock genes. So you see that there's in these heat maps with a Z-core showing the up or down regulation of the different genes, you see a very consistent time-dependent regulation of the expression over time. You see that here. And remarkable, this is actually very similar in the mouse and in the human transcriptome. You see here the comparison. I think this very beautifully illustrates. It's very, very simple. You see that here. So then we did a bioinformatic analysis with 2 excellent bioinformaticians by using dry-R. And we identified there are 1,700 human protein coding rhythmic genes in the hepatocyte. So, and 34%, so this 30% of these human circadian genes are shared with mice. So in this case, there are also distinct differences, and several key genes and biochemical pathways displayed this species-specific temporal rhythmicity. Now, in the following, we only focus on the human hepatocytes engrafted in the mice, which are infected by the virus. So that's possible by basically looking specific at the human RNA. And you see here again, I think you see what's very nicely, you see here cycling genes. And you see which are cycling in the human, they're not cycling in the mouse. We see non-cycling genes in the human, here cycling in the mouse. And we see here, we have the same circadian, the same rhythmicity in human and mouse here, and altered here. So this is I believe is a very beautiful model, not only for hepatitis C virus infection, but also as a general resource to look which genes are rhythmically regulated. And we just presented that at EASL. So what about chronic HCV infection? So we infect now these mice with a patient-derived virus, genotype 1b, then for 8 weeks. And then we do, again, the same experiment and harvest the liver over the 24 period. And here we're starting now with the clock genes. And what we see here that we have the clock genes. We have this rhythm here in the controls, and we see here that the HCV infection changes the rhythmicity of the expression of the clock genes. But what is even more dramatic is then the consequence of these changes on the circadian clock, on the rhythmicity, on the circadian or diurnal transcription of genes in the human liver of this mouse. So this is shown here basically using defining the global rhythmic gene expression in human hepatocytes following HCV infection in this model. You see here the control. So we have in 621 genes, we have a complete loss of the rhythmicity. So here these are the control. The mice, the non-infected mice, the HCV infected mice, complete loss of rhythmicity. We have also genes which are not rhythmic in the controls, but they are rhythmic then in the HCV. We have unchanged genes, and we have altered genes. We have 4 different patterns. So, but what it really shows that the virus changes the circadian, the diurnal rhythmicity of a very large number of genes in humanized mice. So how does that work? And we have more data, but I will not go into detail. So what we also show that the HCV infection alters the diurnal rhythmicity of promoter-associated epigenetic marks. So there, as we showed before, in patients where the virus leaves these epigenetic footprints, and we believe in this case, this epigenetic modifications of the clock genes, but also other genes results in the perturbation. Now, why is this important also for the disease biology? And it's important because the virus changes the rhythmicity of genes, which are driving the disease biology. So, and I've shown you here some examples. We have the fatty acid and bile acids. We have first of all a change in the overall expression level, but we also have a change in the circadian rhythm. So this is basically fatty acids, bile acid. We see SMART, where basically the circadian rhythm is basically blocked. We have a flat line here, and also we have an increased expression, the same for AMT, circadian blocked, and then here for TNF-alpha, NF-kappa B, checkpoints, EGFR targets in terms of innate immunity and also carcinogenesis. And then what is the consequences on the protein expression? And we validated some of the targets. It's technically very difficult. We need to do basically immunohistochemistry to look at specifically the human hepatocytes in the infected mice. And we see here, for example, we can validate here, for example, an upregulation of MYC in the HCV-infected liver. So we believe that these changes that contribute to the liver disease progression in HCV and the cancer risk. Then we asked the question, what about patients? So do we see the same changes in patients? So, and to do that, we compared directly the experiments in the chimeric mice. So these are the different time points. And of course, what we cannot do is there's no data set available. It's not possible to do serial biopsies of patients over a 24-hour period. This is not possible. But we can look at the overall expression of these genes, which are rhythmic, which we have identified as the rhythmic genes. And what we find that the rhythmicity of the expression of the rhythmic genes is similarly perturbed in the chimeric mice as in patients. So this is here the Markus Heim cohort. These are our own cohorts. And what we also found, if we look at patients who have received antiviral treatment with HCV elimination, that some of the genes, they stay perturbed, in particular in patients with advanced liver disease and patients which subsequently develop liver cancer. So this shows that this is clinically relevant in patients, what we see. And then we went one step further. Can we basically use this information to potentially also predict cancer risk? And here we teamed up with Jujin Hoshida in Dallas, who has, as you know, basically has devoted his life on predicting cancer risk with different signatures, developing that also now as a commercial assay. And he basically looked at our data set and looked at the genes which are perturbed by hepatitis C virus infection, defined using this gene set, a so-called HCV clock gene signature. So these are the genes which are perturbed by the virus and persist perturbed in the patients. And then when he looks at these perturbed genes, at basically the clock signature, interestingly nicely correlates with the cancer risk in the patient's cohorts has been studying, and it also correlates here with outcome and liver function parameters as you see here. And finally, actually here I showed the survival curve in these patients. So I think this is very exciting, shows the clinical relevance. So I want to summarize this part. Basically, the HCV disrupted rhythmic pathways in the livers of chimeric mice and patient cohorts reveal a firm link with all major hallmarks of cancer. So this shows basically what we see here in the hallmark of cancers. We basically matched the changes of the rhythmicity of the genes perturbed by hepatitis C. We also show really that the HCV-induced perturbation of the DNA transcriptome correlates with clinical risk of developing HCC, the data from Jujin Hoshida. We see that the HCV-driven deregulation of rhythmic pathways is observed in patients and persists following SVR in patients with advanced liver disease and HCC. And we believe, together with other data sets, this is an opportunity, another opportunity to discover new biomarkers for HCC risk and strategies for chemo prevention. So this is a paper now we have in Nature.com, which is hopefully accepted very soon. So then I want to move on. So having shown hepatitis C as an example of using hepatitis C virus to understand the disease biology, but also an example of reversal of fibrosis, I want to share our approach how to find novel drugs for treatment of liver fibrosis and prevent cancer. And I just show you one example here, and we can discuss about the other compounds we found. So this is actually an old slide. It's for 4 years ago, and I show this on intention to basically look at if all the targets which have been suggested, and which many of them have been actually analyzed in the clinic in many different clinical trials. You see them here. SK1, then you have FXR. We have the CCRs. We have basically LOXL2, and basically, if we are really honest, most of them fail. There's still some hope. I mean, for the FGF targeting agents, I think they look very interesting. Also, PPAR gamma, but I think overall, I think given the investment and the effort in of the industry has been done, it really is quite frustrating in terms of the clinical outcome. So, but there's also hope, and I think one clearly is the resmetirom, which has been approved, and I think here is basically, I think we see progress, especially here on this is a summary of the phase 2 clinical trials, which is now also published. So and basically what we show, we have a very nice effect on the NASH resolution in some patients. Of course, if you look at really the difference between the placebo group and the treated group, it's very low numbers, 16%, and we have a fibrosis improvement if you just simply subtract the two groups, basically at 10 or 12% in that trial. So nevertheless, I believe it's a door opener for me. It reminds me basically of in the early times of hepatitis C drug development, where basically we basically had different compounds as the door opener. I think that's a fantastic achievement. And we also seeing now, we heard a very nice presentation at EASL 2 weeks ago from Boehringer Ingelheim, basically using GLP-1, a dual receptor agonist, glucagon, to show even better results than resmetirom. So I think there's clearly hope. But what you also see, the number of patients really showing a significant improvement of liver fibrosis is still very, very low. And I think we have to see in the other compounds. I believe this is really remains a major challenge. We need the combination, I believe, of a metabolic MASH drug, and but also having an antifibrotic drug. So why is it so difficult? Why has industry failed, and also academia failed really to develop efficient drugs for advanced liver fibrosis and cancer prevention? So one is that we have really complex cell circuits mediating pathogenesis. It's not like one simple target to get the trick. We have multiple cell types, including liver non-parenchymal cells. The hierarchy of the pathogenic mediators is poorly understood, so we still don't know what's the driver, what's the bystander, what comes first, what comes at the end of the cascade. The other problem is that current animal models only partially recapitulate the human disease. We've seen this. Many drugs are fantastic in the CCL4 model, the chemical models, and at the end, they don't do anything in the patients. We don't have any. Still, I believe there was a very nice paper, you all have seen that in Nature Metabolism, ranking the different models. But still, I believe it is very a challenge to predict translatability to the clinic. So what we need is, and this was a driver for our research, we need new models recapitulating the key circuits of patients' disease, and then exploit these models to understand the disease biology, and also then define new targets and drugs. So what we did, I did the sabbatical at the Broad. I met at that time Yujin Mushida was there, where we just left the Broad, and he was working with Todd Gollob at that time, and he published in the New England Journal a paper just 2 or 3 years ago, and he had in 2008 identifying a prognostic liver signature, which very nicely distinguished patients with stable disease, with what he calls good prognosis, and patients with poor prognosis, which were characterized by HCC development, decompensation, need for liver transplantation, or death. And he, in the meantime, reproduced this, and others, Ray Chung, others reproduced the impact of this liver-based signature in many, many different other cohorts, including ourselves. We saw that in retrospective analysis. And if you look at the genes which are in this 186 gene signatures, we have the poor prognosis genes. So if they are upregulated, basically the patient has a bad prognosis, interferon, IL-6, TNF-alpha, NF-κB, TGF-beta, stellate cells. So I think that's the usual suspects. And then if we have a good prognosis, we have basically the metabolic genes and the bile acid metabolism genes. They are upregulated, so the liver is healthy and is able to function well. And here we have also an upregulation of the DNA damage repair. So basically, this could be also one reason for the decreased cancer risk. So then we were able to model this signature, which takes 20 years in patients, in less than 20 days in a cell-based model system. So this made us really very excited. So in contrast to previous models, we believe this is clinically relevant because it is really the endpoint is really a clinical signature. It's a multi-target approach. It's not like a single target. So we can basically assess the effect on this 186 genes. And what we then did, we applied this model to identify compounds and targets to revert the PLS for inhibition of fibrotic disease and progression, reduction to HCC risk, and improved survival. So basically, that's what we see, what this signature predicts. And this is a paper we published in Nature Communications. Just want to walk you through some of the key results. How do we do this? So we started with hepatitis C virus infection. So we used a co-culture model with LX2 stellate cells, and then also we added macrophages. And then these are differentiated HLA-7 cells. They're very similar to, or partially similar to human hepatocytes, where it's a system, a high throughput system. And then if we infect these cells with hepatitis C virus, we actually induce the poor prognosis status of the signature, which predicts death and HCC risk and poor survival. This is shown here. We see this on the RNA level, but also here on the protein level. We see this is actually specific for the viruses causing liver disease and cancer, HBV, HCV. The dengue virus does not do it. It causes acute liver injury, also alcohol, free fatty acid. But for example, another cell stressor as low glucose does not induce the poor prognosis status of the signature. So in orange, poor prognosis, green, good prognosis, and here up and down regulation of the poor prognosis and the good prognosis genes. So the poor prognosis liver PLS induction is specific for the major etiologic agents responsible for the fibrogenic carcinogenic liver disease in patients, meaning the two viruses, alcohol and free fatty as a model for MASH. So how similar is that to patients? So that's always a question if you have a cell culture system. Here, Jujin Hoshida did a detailed analysis, and what is really very nice that we find a very nice correlation here in patients with the different disease etiologies. So what's also very interesting, since we used a triple culture system and also basically a very special cell culture system, we were only able to model cell circuits not only occurring in the epithelial cells but also in the non-parenchymal cells, which is very important because we all agree that the non-parenchymal cells, the microenvironment of the liver is key for the disease biology and also for drug development. So what we then did with Jujin Hoshida, we did an in silico screen first. So basically, we're using the LINCS and CMAP databases at the NIH and at the Broad Institute, and these are databases which now actually have, in the meantime, more than 50,000 compounds and also CRISPR RNAi screens, which basically give the information about what this drug or the knockout does to the gene expression in different cell lines. So this allows us to identify in silico candidate compounds and candidate targets which will revert the signature. So we found from this screen, we found 25 candidate bioactive compounds. We assessed them in the cell-based system, And what we found is in the screen, these are here the top hits. So we found captopril, which is an ACE emitter, as you know very well, which actually we know for a long time that ACE inhibitors, the ACE or RAS inhibitors, they are antifibrotic, but they're rarely used due to the low blood pressure, which is fully tolerated in patients with advanced fibrosis or liver cirrhosis. But we found another compound which was a big surprise, and this was nizatidine. Nizatidine is a blocker of the histamine receptor 2, and as you know, maybe the physicians many years ago, this was one of the first standard of cares to treat gastric ulcers and GERD. So what is the histamine 2 receptor? It's a G protein coupled cell surface receptor expressed in several liver tissues, including the liver. And what we found very interesting, that this is a completely different and complementary to compounds in clinical development. So it's really something out of the box. How does that work? And so here we then did proof of concept studies in two models. So this is the DN cirrhosis rat model. This was done by Brian Fuchs at the Harvard Medical School, who's now in industry. And Brian Fuchs actually compared this compound to all the other compounds he tested, including the ones which are now approved. And he always said this is a fantastic drug. It's basically at least as good as the drugs in clinical development, or actually the approved drug, resmetirom. And so here you see the effect on the collagen proportional area, hydroxyproline, the fibrosis markers, and we see here, I think, a very significant inhibition of the liver cancer in this choline deficient high-fat diet model. And we see the same actually here in a mouse. So this is basically, and here he looked also at the, here we were looking also at the tumor nodules, and we see here a very significant reduction of the tumor nodules. So this actually here, this is here the mouse model. We used the mouse model where we basically also, we injected the mice with DN, we fed them with a choline deficient high-fat diet. The mice developed fibrosis and HCC-like lesions, and what you see here, this marked reduction of fibrosis and cancer. So really stunning proof of concept, very robust. Then the question is, how does that work? So that's a completely novel mechanism. So we then looked at the expression of the target, and at that time we were very happy that we had just 2 years before we published our paper in Nature on the first human liver cell single cell atlas. And so looking at our atlases, but also on other atlases, we found that the histamine receptor actually has the highest expression, not in the hepatocytes, but you see here in the macrophages. So what is the mechanism of action? So here we basically further zoomed in which are the macrophages which express the histamine receptor. That's the beauty of single-cell RNA-seq, which we are all using now. At that time, this was really something completely new and exciting. And then we see that the histamine receptor is highly expressed in a subset of pro-inflammatory liver macrophages, the CLEC5I, hey, Marco macrophages. So how can we prove that the macrophages are really the nizatidine target? So here again, we used single-cell RNA-seq, and we combined it with a spheroid system we had established in the lab. So basically here we have resected liver tissue. We dissociate the tissues, put them back again into tissue, and then incubate them with nizatidine, and then look at the effects on the cell phenotype. So, and what we found here, basically in here we are focusing on the immune cells. We know these contain the macrophages. They also have telomerocytes and other cells, and we see here again the expression of the histamine receptor in these macrophages. And if we look at what does nizatidine do in the macrophages, we can then basically, using the single cell analysis, we can look at each single cell and check in which cells we see the effect and what is the effect. And what we found is that nizatidine treatment results in a marked shift within the liver transcriptome. The majority of these macrophages are pro-inflammatory, and it induces a shift from the pro-inflammatory, pro-fibrogenic, atypical immunoregulatory macrophage. And we actually then could basically validate it in vivo experiments with Matthias Heikenwälder here at that time. And what we see that the Nizetidine improves the liver inflammation and immune surveillance in vivo by recruiting then also T cells and changing the macrophage content of the liver. And so that's how we think how that works. So the Nizetidine basically acts on them. We have here the macrophage, which results in activation recruitment of neutrophils, hepatocytes, T lymphocytes induced by the chronic liver injury. And then the Nizetidine or H-histamine targeting compounds provide a therapeutic candidate approach for patients with fibrotic liver disease by modulation of the secretome, then improving the anti-cancer immune responses, improvement of liver fibrosis, and cancer prevention. And so basically what we have shown here, we have developed a fast, robust, and clinically relevant cell-based model system, modeling the PLS in cell culture, which allowed us to discover novel compounds. I've showed you Nizetidine. We have also several other compounds and targets which we are currently working on, including anti-Claudin antibody. Perturbation studies combined with RNA-seq unravels the mechanism of action, and the PLS system combined with single-cell RNA-seq provides a robust, fast, and simple platform for liver disease and drug discovery. You see that here from the drug screening in the PLS validation, basic proof of concept in animal models, single-cell RNA-seq for the mechanism of action. And I want to use the last couple of minutes to show you now data which we presented at EASL 10 days ago, and that's basically now how do we translate this approach now into the clinic. Our goal, of course, is to we believe to perform a clinical trial because this is a very cheap drug. It's a generic drug, so this is not something industry will quickly develop, but it's something I think especially for academia and also I mean the need for having a low cost and very simple and safe approach. I believe this is a fantastic compound. So what be a data set to convince us and also funders like, for example, the NIH, they are interested or INSERM to do a clinical trial. So what we did first, we looked in the target expression in patients, performed additional proof of concept studies in clinically relevant mouse models, and looked for a biomarker at the end to stratify the patients and also to at the end see a first response without the need of a biopsy. So and just I want to really work very quickly because we are almost at the end of the presentation. So I mean, being a founder of a company which is now clinical stage and talking also to investors and with, I think, what's a good compound? And I think the first thing you want to show is to show overexpression of your target. You want to have a nice proof of concept in clinically relevant models. And then you need a biomarker to test target engagement and show efficacy in a phase 1b trial or phase 2a trial without need of biopsies, and just go through that. So we see the histamine receptor is overexpressed in liver disease with HCC risk, and we see also the expression of the receptor level is associated with survival, as you see here on the right side. So these are data from Juji and Hoshida. And then we teamed up with Ray Chung at Harvard, who's actually very interested in this project. They actually, and Ray and Juji, they're actually doing a chemoprevention trial with statins at the moment. And here you basically, Ray asked a fellow of his, Jun Song, to basically look in the data of the National Health and Nutrition Examination Survey 3. So this was a large cohort of 33,000 patients at that time, and this was also the time when Nizetidine was quite frequently used. And what they found actually that the use of Nizetidine and Ranitidine, which is very similar, was associated with the non-invasive measures of liver fibrosis. So this is just an association. It's still a low number here of patients who actually then took the drug. But I think it comforts very nicely that there's a signal in a very well characterized patient cohort. And then we basically did some additional experiments in patient-derived models to convince ourselves that this is something translatable. And what we did is here we have developed over the last couple of years a spheroid model. So this is a model where we take the patient tissue from either healthy or diseased mesh of fibrosis patients, We dissociate the cells, we grow them then in a medium enriched with growth factors, we then grow patient cells, and the beauty is they contain all the NPC cells. So this is basically, these are not organoids. So these are really it's a cell-based system which contains the endothelial cells, the Kupffer cells, the stellate cells, contains also CD4 and CD8 cells. We have a paper in JHEP reports coming up describing this system. So then basically we use this system to look at the prognostic liver signature. We induce the signature, this poor prognosis signatures in patients with free fatty acid, and then if we incubate them, these cells within nizatidine, we see the reversal of the signature. It does not work in all patients. You see that it's patient specific. So not all patients have this nice reversal. So this is a system also which would allow us to test that. And then also we see here if we can also induce the signature with histamine. And what we also basically then showed that the expression of the target, so the higher the nizatidine level is in these patients, the better is the response. So this is very important, again, for the basically the clinical translation that we see the target expression is associated also with the effect on the prognostic liver signature. Then very quickly, we then used the human liver chimeric mouse model as another model, which we believe is much more translatable than the classical model. So here we engraft FRG not mice with human hepatocytes, and then we feed these mice with a colon-deficient high-fat diet. These mice then they develop liver fibrosis and tumor nodules, HCC-like lesions. And what we did, this is only a small group. It's very difficult to generate these mice. You've seen them also for the HCV model. So we have only small numbers, so that's why it's hard to really get very high significance. But what we see a very nice trend here for the inhibition of fibrosis in the collocation proportionate area and also in the tumor models in this system. And finally, we did what is the ultimate proof is the genetic knockout. So the loss of function, we knocked out the histamine receptor. That's what usually industry wants to see before moving forward. And we actually showed again that if we knock out the histamine receptor, we have in a mesh model with colon-deficient high-fat diet, we see a massive reduction of the tumor carcinogenesis here. And so how can we, and with these slides I want to finish, how can we now basically develop a biomarker for these patients, which would us to allow for a clinical trial to show whether this compound has really clinically an effect, because that's what we want to know before we, let's say, biopsy the patients or have a very long-term study to look at HCC development. And here again, we use the prognostic liver signature. And Jujin Yoshida, in the meantime, has developed a serum-based prognostic liver signature, which he calls PLS-SEC. So this has HCC-associated risk proteins and alpha-fetoprotein. You see that here. And what you see here is that these blood-based PLS-SEC AFP signature accurately stratifies patients with advanced liver fibrosis for long-term HCC risk and predict recurrence risk. So he has published this in Med and another paper in Science Translational Medicine. And so we then use this blood-based prognostic liver signature, the PLS-SEC, and checked in the humanized mice and the HCC rats whether here you see the proteins, CCL27, IGF-BP7, MMP7, IL-6, VCAM1, and these two others as high-risk and low-risk proteins. And we're actually using the ELISA he has developed and actually show that the nizatidine it reduces the PLS score. So I think this shows that it validates the PLS-SEC as a candidate non-invasive biomarker for a future clinical trial. So this is my last summary slide. So summary of the histamine receptor 2 inhibition. We have the loss of function showing that the histamine receptor is a driver of hepatocarcinogenesis in a MASH model. Nizatidine inhibits fibrosis progression and tumor formation in a human liver chimeric MASH models, but also in the classical RET-DN or the DN combined colon-deficient MASH HCC mouse models. We see an improvement of liver disease associated with PLS-SEC reversal associated with HCC risk in the serum of treated mice. And we conclude that the low cost and very safe histamine receptor 2 antagonist nizatidine is a candidate compound for treatment of advanced liver fibrosis and HCC chemoprevention. we believe that the treatment-induced reversal of clinically validated PLS offers a very simple, non-invasive biomarker as a readout. And what we hope is that this results pave the way now for the clinical development of histamine 2 receptors for HCC chemo prevention using this as a clinical readout. And to close, I want to thank all the collaborators here. This is our team at our institute, but also the University of Strasbourg, the Strasbourg University Hospitals, in particular Naoto Fujiwara, Yujin Hoshida, Dominique Grün, Single Cell, Matthias Heikenwälder, Nathalie Pochy, who's now heading Genentech R&D, Ave Frege, who is heading now Genentech R&D, and then Ray Chang, Brian Fuchs, Nabil Badisi at the Mass General Hospital and Harvard. And I thank you very much for your attention. Yeah, no, that's a very good question. So that's a paper we have in revision at the moment. So basically we have, so what has been shown is that actually targeting the circadian clock improves the metabolism. So there's a couple of papers on that, and there's also good evidence from clinical studies, target expression. We wanted to know specifically also, is also fibrosis targeted as a consequence of metabolism. So what we did is different approaches using different mouse models and then using single-cell RNA-seq, or then isolate. We used, again, our workhorses for fibrosis is choline-deficient high-fat diet because it's rapidly inducing significant fibrosis. And then what we did is we again did a time course experiment, and then we isolated the stellate cells basically from the livers. And this allowed us to specifically look into the stellate cells rhythmicity. And what's interesting is that the stellate cells also have a circadian clock. They also, it drives actually the expression of the profibrotic genes. And so meaning if you have a perturbation of the circadian clock, either by the high fat, by the diet, but you can also have that, for example, by travel. As you know, the jet lag is very bad, and as you know, there's clinical data that basically people who are traveling a lot with jet lag also, or the shift workers, they have a higher risk of cancer, so especially breast cancer, but also liver disease and liver cancer. So I think this is there's clearly very good epidemiological data showing that the perturbation of the circadian clock results also in liver disease and cancer. And we believe one is the hepatocytes, which has been shown by others, but also the stellate cells both play an important role here. It's an excellent question. We have not looked at that. We have in the study before, we did publish in Gastro and Gut, basically what is possible using an epigenetic inhibitor, like say targeting bromodomains. JAK1 we use as an experimental drug. We can inhibit the epigenetic changes, and then we improve also outcome. We have less fibrosis, we have less cancer in mouse models. We have not yet done that in the HCV study. We have not. It's a very good question actually. How can you target it? And which would be the drugs, how to target it. But these are definitely on the list, that's for sure. So I think it's a very good question. So we looked at different pathways, and I showed you, for example, SMART, but also other pathways. And here again, the rhythmicity stays perturbed also in post-HCV cure. And in patients, we only can measure the absolute levels. So we cannot measure because we don't have serial biopsies where we can see whether the absolute levels of these genes remain perturbed. So we believe it has a clinical relevance, and we see again, like we see what we did before the epigenetic change. This is in particular the case in the patients with advanced liver disease and patients who are developing then HCC also. That's very good. I think that's a very question. Absolutely. I mean, the earlier you start, I think the better is the effect. I agree. Regarding the residents, so basically what we did, these were basically the studies using single-cell RNA-seq is basically we used liver tissues from the patient. So these were really macrophages within the liver, resident macrophages within the liver. So we take a liver tissue, we dissect it, and then we put it back and grow it as a liver spheroid.
Okay.
And then basically having this liver spheroid, and then we add the compound for 3 days or up to 10 days. And then we perform single-cell RNA-seq and check what is the compound doing on each individual cell in the liver sphere rate. So this allows us to really look at the cells. And the beauty is it's really cells, it's only cells in the liver. We don't add anything artificial. The model system in the beginning, this is a cell-based system, but then for the validation, we use really the authentic patient liver tissue. So that's why we are very confident that this applies for patients.
Well.
It's a very good question. These are basically standard mice we obtained them from Jackson Labs, which basically has a knockout, an ubiquitous knockout.
Ubiquitous. Okay. So there is no, you cannot make the knockout, for example, in CLEC5.
You could do that, but we didn't do that so far. For us, okay. It was for two reasons we did the experiments. One, to basically have the genetic validation. That's always industry, but also FDA likes that if you basically show that there's a clear validation. And then the other point is, what's very important is the safety. So if you knock it out, there's basically no major adverse effect, because that's the other danger. Let's say if you add the compound and you would basically, these mice were dying or these mice were very sick, have, let's say, whatever, lung disease or so, then you would also worry about long-term effects of nizatidine. And this is an experiment. On one hand, it confirms the target, but also reassures the safety to target this pathway in that disease setting, because then we injected the mice with DEN, we gave them the colon-deficient high-fat diet, so they developed advanced liver disease in that system, and without any detectable major side effects, which is very important. It's a good question. We have not yet. It's a good question. We can look at that. We did not. I don't have to check again whether we did that we did a whole panel, but I don't remember. I think if we did, there was no major change. But it's important. I agree. We have to look at this. But still in patients, it's I think not for nizatidine. I'm not sure. Maybe it's the case, but I've not seen any really hard data that there's really an issue on the glucose metabolism. Very good. That's the next question. I mean, they're less used now. We have basically the next generation of basically omeprazole and the proton pump inhibitors. But I think still we have. So I think it's what we see is basically we see nizatidine, ranitidine. We also see difference. We actually compared different H2 blockers, and we found actually nizatidine has the strongest effect. So which could be related to the structure. It's maybe not only the binding, but the structure. So we think it's really related to the histamine receptor because of the knockout system. So it's not an off-target effect. But basically that's also something to think about. What would be the best drug to take out of the class? From our cell-based data and animal data, we think that it's the nizatidine to start with.
Excellent, Thomas. So we are right on time. I think it's 5:02, so we will close here the session for today. I just want to thank you and thank the audience for listening and attending today to this great seminar. I wish you a very nice holidays on behalf of the liver seminars, on behalf of Jordi Gracia Sancho and Rafael Bañares and myself. And I hope that you will join us in the next session after the summer holidays. Thank you very much to everyone.
And thank you very much for the kind invitation. Really fantastic and a wonderful discussion. Really
thank you, Thomas. Thank you for attending. Thank you.